Go ahead.
Okay, I just wanted to ask about the RBC.
Okay.
Okay, if you want to increase RBC,
Yeah.
okay, you told us that we need to remove our blood and keep it in a refrigerator.
Yeah. No,
Okay.
it's a simple blood transfusion.
Yeah. Okay. Yes, sir, I didn't get you.
Okay,
Hello, sir.
I will explain that. Okay. How the initial blood doping started is that, say now you all know about the blood bank. Okay.
Yes.
So normally we go give blood. I think we have the ABO group, and they will store it for a couple of days because it is good. Then if some patient requires, it will be given. I think you know about the blood transfusion.
Yes, sir.
Okay. Now, in case of a blood doping, so how are we going to increase the RBC concentration in a sports person? Okay. We draw blood from a person. Suppose if I'm competing in Tour de France, they draw blood from me. They store it in the refrigerator. So once they draw blood from that, now my body feels that I think there is less of a blood. Then they started making more of RBCs. I will not be anemic, but I'll be doing all the activities. But some blood has been taken away. But during these 5 days, some blood is going to be synthesized more. All right. Then the same blood is going to be transferred back to me so that I will have an additional RBCs with that one. So that I will have more RBCs, then more oxygen to supply to the muscle cell for the endurance. In fact, that is how it's been done.
Okay, sir. Thank you.
Good. So my next topic is similar related to that. Yeah, go ahead, sir.
How many days before they are taken the?
It's a 1 week, 1 week time, 1 week, because you cannot keep blood for more than a week. We cannot keep the blood for more than a week. So they have taken the blood. So then in 1 week, I think you will develop all these stuff, and then it is given back. But now now the blood doping is not there, basically because it requires transfusion at the time. Now they give a hormone. Erythropoietin is a hormone which synthesizes RBC cells. So that cannot be detected. So even erythropoietin administration is very bad. But in a clinical conditions, if somebody is having a severe anemia, then to such people erythropoietin is administered, but not to a normal healthy person.
I think it spoiled the kidney, sir.
Usually erythropoietin is released from the kidney. Suppose if kidney has got a disorder, that's where the anemia is going to come. For example, if kidney is not functioning, if there is a kidney failure situation, then depending upon which region, if the erythropoietin is released, then automatically they will become anemic. Suppose if you do have a kidney disorder, then we cannot give protein-rich diet because the degraded amino acids has to be filtered, which is going to be very difficult, isn't it? So they will become really, really anemic. So during that particular time, an external administration of erythropoietin is given.
Thank you, sir.
All right, I really enjoyed the discussion, and I'm also, like all the questions. So that means I think I have, at one level, I have communicated something to all of you. Let's see like how my other topic will get into this. Probably this is, many of the people also ask the question whether it is ethically right or wrong. But we will just discuss the possible things, how it can be done, or which is ethical or which is not ethical. Now the question is for physical education teachers, I think I don't have to give any introduction about the methodology and the process which is going to be how the talent identification in sports and games. That's a very critical event. In fact, if you do identify a person, he is good in dancing, good in singing, good in running, so then we can mold them and then we can try to get the best out of them. So usually I think you all know that between Lata Mangeshkar and Sachin Tendulkar, suppose if Sachin Tendulkar has been given the training for singing, whether he would have been like that, or Lata Mangeshkar with cricket. I think we all know about those analogies, in fact. So we will try to make a similar analysis, whether can we identify the talent, that is through a genetic analysis. So now I think you all know about the National Association of Physical Education and Sports Sciences, NAPIS, and Professor Chandrakumar happens to be the member of that. And we published a research article. The NAPIS has got an online journal. Now that is the prediction of high performance in sports and games based on genetic analysis. That paper is online, is available. And whatever I am going to discuss today is the information we got it from this, in fact. So here we also wrote a research project, myself, Professor Madhyalagan and Chandrakumar. But getting funds for something like that is going to be quite hard. But you people has to, in future, you have to write grant and get this particular information. So prediction of high performance in sports and games based on genetic analysis. Whatever these slides have been prepared is together with Professor Chandrakumar and myself. It is not that I only did it. I think it's a combination of both Professor Chandrakumar and so one of my MSc student, Rushali Kamath, I think she has compiled all the data, whatever the information we asked. She bought those informations and then we can compile that into a form of a published article, in fact. I think you all know about this Kambala runner. And I think it's been also related that I think he ran faster than Usain Bolt. If you just calculate, because the distance in case of a Kambala is 145 meters, I think he cleared that in 13.62 seconds. If you recalculate for 100 meters, it is like 9.55. So now when somebody has given this particular information, I think he got into the limelight and many people started felicitating him. So this is the Kambala sports, in fact. Then everybody said, yeah, we have a raw talent, we have to identify, we have to nurture. This is how the whole process started now, in fact. I think all the people are felicitating. That's good. I mean, recognition in a particular sport, especially from a Kambala is a native sport. Not many people know about it. But we do have really, really raw talent there, in fact. How do we identify them? How do we nurture them, in fact? This is Professor, our CM Yediyurappa is felicitating. The Sports Authority of India is felicitating. Then immediately there was one more. So that is the Nishant Shetty. He's a 20-year-old Kambala jockey from Karnataka. He broke that record and his is something around like 9.51 seconds, in fact. Then when these people are started comparing for Usain Bolt, so then came the press conference, that is the Ashwini Nachappa. He says, it is unfair that I think these are all two different events. I think we should not compare. We should not put any pressure on any of these people so that, okay, come, you run in front of us in the track event. We will clock the whole process. He said that is unfair. And now the question is, we have to discuss whether at what level now, whether Nitin Shetty or these people who have done the Kambala race. So can we take them? Okay, no, these are all talented people. We will train them. Or can we identify such talents in a very early age so that we can train them, so that we can get best out of it, in fact. So now when it comes to the talent identification, for example, usually there is a raw talent. I mean, you cannot just ask everyone to do all kinds. Suppose if they ask me to do, because I love to play shuttle badminton, but if you ask them to run, I think for me it's going to be very difficult. So as a result, many champions are born with inherited physical potential, and by providing all kinds of support like technical, psychological, environmental, they can be molded into top-class performance. Individuals have always been screened through different means for their athletic, physical, and behavioral proficiency and contributes to their high performance in sport. For example, when you do, a coaching camp, so there are about like 20 people, you will identify quickly that guy is good. And for example, so you are put into some rigorous training, and probably bulk of them tomorrow they can't come, but a couple of them show up. Sir, we are ready. We want to do something. So you will find some enthusiasm in some people, some raw talent really in the early stages, in fact. So you have a methodology. I think what you call this pre-participation physical evaluation, PPE. So to screen for illness, injury, susceptibility, or medical conditions, counseling is always being done to understand the psychological aspect of an athlete. I think this is being mandatory for an administrative requirement in your case. However, the main drawback to these screening methods have always been that it was time consuming, non-uniformity of protocols, compliance issues, and occasionally no follow-up is done. You identify someone, we don't know, like, how they. So with all these existing conventional screening procedures, if the athlete does not possess the required inherited potentials, any form of support will result in identifying or developing an average athlete. So we want to have, because this time the Tokyo Olympic did not happen because of the corona pandemic. So during the Olympic time, I think everybody will discuss this. Ours is a 1.3 billion population. We don't have a single person to getting a gold medal in athletic event, a game event. So during that time, I think we are going to hear a lot more such discussion, in fact. What are the alternate methods available? Is there any other way a standardized protocol could be established for the identification of, for example, sports high performance athletes that can, or it can screen any prevailing disease conditions, whether it is accurate, practical, cost effective, and also be in, even if there is some deformities, I think it is treatable. So one more thing is that what I also found out that, I don't know, the sports person are really afraid to give blood, because myself and Professor Chandra Kumar, along with Professor Venkatesh Kumar, we do have a small research project. So we talk about the stress related to the free radicals and then the damage related to the free radicals impact. So we asked a couple of Maharajas College students, so why don't you give a 2 ml of blood sample? They were all scared. I don't know, because all these blood samples is going to be considered to be as an invasive. But when it comes to the genetic analysis, so what we are going to use is a non-invasive sample, in fact. We will just discuss what exactly is that. So the answer what we are discussing right now is the genome analysis. It is an integral part of molecular biology. So here, a simple saliva sample or a cheek swab can donate the whole genetic blueprint of a person, in fact. So here, where basic molecular technique of extraction of DNA, genomic studies is going to be done. Sports genomic is now a new upcoming field that is gaining momentum in the world of analysis and improvement of sports high performance. Uses the genetic knowledge of the person to assess their abilities and performance in sports. I think I can best explain in the current pandemic, COVID-19. I think everybody knows about COVID-19. I think even in the news, you all have heard an RT-PCR, in fact, all right? So RT-PCR literally means it is a reverse transcribed polymerase chain reaction. I will not go in detail, but at least I will explain. It is easy for me to explain some of the cellular events. And the test is non-invasive, basically because for corona test, they are going to take a swab either from the nasal or from the throat. It is not a blood sample. So anything which you are going to prick, it is an invasive. For example, if you go to a cath lab, cataract, I mean for angioplasty, all those things are all an invasive procedure. Colonoscopy, endoscopy, they are all invasive. But now here it is a non-invasive, in which you are going to take a nasal swab or a throat swab, because this is exactly what is being done for RT-PCR. To know the genome of a sports person, what is required is that few cells isolated from the saliva. All we want is that he has to just give the saliva, 5 ml, 10 ml, because usually the saliva comes up with some of the epithelial cells. Those cells are very important. I think the RT-PCR, I can also give an example, is what is known as, there is a technology called fingerprinting, fingerprint technology. So usually what happens in case of a crime scene. So in a crime scene, I think if there is a blood sample, so now the question is, you suspect about 10 people are involved in this murder of a person. So now the fingerprinting with a blood sample or with a hair follicle, so we can easily identify the whole thing in fact. So that is the accuracy what we are going to get it, the genetic blueprint in fact. So now, so this is an example in order to explain this genomic analysis. What we require is a non-invasive methodology, and then all we need is a couple of cells from the saliva that is good enough so that we can do the genome analysis. So in Mysore, we have an institution called Anthropology Survey of India. Okay, so now what is the main objective of this Anthropology Survey of India is that because right now the human genome project is over, because all over the world, all countries, many institutions, universities, laboratories, they work together for more than 13 years to identify the blueprint of a human being. So that one is out now, because now the human genome is out. Now from this human genome, now we wanted to see if there are any basic differences. What we call this as a polymorphism. For example, so we do have parents and then we do have the children. The children will inherit some characters of a father, some characters of a mother. So you can identify that there is unique character, but you also identify there are some characters inherited from father or mother. So such differences can be easily characterized by the DNA analysis. Now the Anthropology Survey of India is doing the map of several tribes. Just to name, in Biligiri Rangan Hill, there is a tribe called Soligas. Those Soligas, they have a very genetic disorder, what is called this as a sickle cell anemia. Okay, I'm going to discuss the sickle cell anemia in due course. So basically because their blood RBCs carrying capacity of oxygen is very bad. Okay, suppose like if they are getting into higher elevation where the oxygen is going to be in less concentration, for them the breathing is going to be very difficult in fact. So now this sickle cell anemia is a genetic disorder in which if parents have it, the children will also have it in fact. So by analyzing the whole process, they are trying to find out in some advantages also are there, because some tribes will never get diabetes. We want to find out why and how. Or some tribe, I mean, they are not exposed or they don't get any arthritic conditions in fact. Why is that? So what is the difference in the genetic makeup? These are the things which is going to be done. So we also have All India Institute of Speech and Hearing, AYUSH. So there also we have a lot more inborn complications. People cannot speak, people cannot talk, they cannot hear. So what we call this as an autism, dyslexia. In university, we also have a genetics and genome department in fact. They are collaborating with the AYUSH and then try to understand the genetic map of autism, dyslexia, then with specialized training, so we can improve them in fact. So now the Anthropology Survey of India is also doing what is known as a Mysore family study. So basically they wanted to find out whether the diabetes is hereditary or not. So what I want to give you the information is that this blueprint will tell us what the nature of a person is that. Okay, now in a group of, I would say 20 students, if somebody says, okay, the person is very good in enduring sports or power or concentration. Now, if you try to get the genetic map, whether is there a difference between a person who is so calm and the person with an endurance, then we can train those people accordingly is the whole concept. But this has got a lot of ethical issues are also there in fact. Whatever we are going to do, this, I think we have to take it with a pinch of salt, that it has to be extremely carefully, we have to view this whole process. I think there is no one correct model for talent identification and selection. It is dependent on the complexity of the sport from different kinds and the infrastructure of the sport at a local, regional and national level in fact. So now the question is, when it comes to the physical education, the main focus is always on best performance and honing of skills and abilities. The primary aim is always the well-being of the athlete. Okay, so now what are the factors which affects the sports? We have training methods, physiology of a person, anthropometry, psychology, biomechanics, nutrition. But the major component which is being neglected happens to be the genetics. The 66% of the ability lies in the blueprint of a person, in fact. So since I would like to concentrate on that one, in fact, I think this is an information coming from the sports genomics, that 66% is responsible if an athlete ability is depending upon the genetic component of it, in fact. So now athlete status is now found to be the heritable trait, and on an average 66% of variance in athlete status is explained by the genetic factors. Its development is later done by the environmental factors, whether you are in the hilly region, you are in the seashore. So there are multitudes of factors that are responsible for elite athlete performance. That is a combination of physiological and psychological factors, and they can be measured by mapping the genetic markers. So what are the traits we are going to analyze? Maximum oxygen uptake, skeletal muscle compositions. I think even in case of a fat, we have a brown fat and then we have a white fat. So depending upon the compositions we are going to talk about, then the motor activity and also the maximum aerobic performance, in fact. So performance variants can be found in differences of lifestyles, environment conditions like terrain, distance from a sea level, eating habits, motivation, sports equipment and training programs. All these contained within the genetic material of an individual. So this is the next step of enhancing athletic performance, and it is done in many developed nations already. It is high time this concept was incorporated in the Indian sports field to generate elite athletes more effectively. Because more than that, I think whether it is good to do such type of study is something which is you also have to be think in this line. So now this is my student has done this particular slide. Imagine you are coaching 5 children. How much average time would it take for you to understand the talent of those kids? What if there was a method to understand the children's propensity for power, endurance, speed, etc., just by looking at a sheet of paper, even before you met them, good or bad? If one sample of saliva could tell you that a child has good strength but poor endurance, that will definitely benefit a coaching strategy. So imagine one child has a symptomatic disease that the parents do not know about, that manifests only in high-level training conditions and can be fatal. Would you want to be notified beforehand? Because some children will also have inherited diseases, in fact, which is also going to be useful. Because such studies has been demonstrated in US, which I will take few examples in that case. I think if it is too technical, you just forgive me. Now, this is how the chromosome look like, and this is the extended version. In fact, now there's a human, we have a cell, in the cell we have a nucleus, in the nucleus we have a chromosome, then the chromosome is made out of what is known as a DNA, that is deoxyribose nucleic acid, in fact. Now, these DNA do contain the information. For example, the DNA of a mosquito or the DNA of an element or of an elephant determines the size and shape of it, in fact. Simultaneously, the way in which all these DNA and the genes are arranged, so they are responsible for all of us. In case of a human being, we do have 22 pairs of chromosomes. These chromosomes, we call this as autosome. And then we have one more pair called X and Y. We call this as a sex chromosome. And we have a pair, means one chromosome, we got it from mother at the maternal side, and another chromosome from the paternal side, in fact. And when you just look into the chromosome, I think for our study of genes, so we are going to classify that there is a centromere, then there is a one arm on top, one arm on the below, we call it a P arm, and then the Q arm, then again there is a subdivision. This is done basically because if there is a gene for a muscle, we want to find out where is that gene is located. It is not located in all chromosomes. It is located in probably chromosome number 8. In that case, whether it is in the upper region or in what region. So in order to clearly say the location of a gene, I think we are going to structurally organize how these chromosomes are going to be there. So we have numbered the chromosome 1, 2, 3, 4, 5, then 22 pair. And whether to determine a boy or a girl, we have X and Y or XX chromosomes. So these are the sex chromosomes. These are the autosomes, in fact. So now, whatever the characters we are located in this 23. So this is a different pictures in that case. All right. So now here what happens is that how the information is going to be transformed from one person to another person. So what we call this as the central dogma of a life. Okay. I also discussed about RT-PCR, reverse transcribed PCR. Now the question is here, how the information is transformed. For example, my RBC is good or bad. When the RBCs is going to be prepared, how these RBCs are going to be done. The information is always carried. We have a nucleic acid, that is the DNA. The information from this DNA will be transferred to RNA. So that particular process is called as a transcription. Okay. From the RNA, protein is going to be synthesized. What we call this as a translation. So now earlier, how the information is going to be transferred from one cell to another or from one person to one generation to next generation. So we have a transcription and we have a translation. So DNA will make RNA, RNA will make protein. Now when you come to the coronavirus, okay, because viruses are all going to be the very primitive one. All right. Now, when you do talk about DNA, RNA, and then the protein, then the genetic component has to be a DNA. But when we try to isolate, characterize virus, then they found out that many viruses, their genetic component is RNA, but not DNA. So in that case, so from RNA, are we going to get protein? No. They say from RNA, we are going to have the DNA. From the DNA, again, we get RNA and protein. So earlier, whatever we thought from DNA to RNA, we call this transcription. But once we started identified that there are viruses which is also made out of RNA, then RNA has to be first converted into DNA. So now that is called as a reverse transcription, in fact. So the coronavirus is made out of RNA. So our RT-PCR, what it does is that first RNA will be taken up, it will be converted into DNA by a reverse transcription. So whatever we talk about the RT is nothing but that. So this is the central dogma, which since the COVID is going to be there, I just wanted to discuss about like what is a transcription and what is a reverse transcription. So RT-PCR is nothing but RNA is first converted into DNA, and then we are going to have all the other process, in fact. So now, where exactly we do have internal cell and we have several components. So now we have the nucleus. In the nucleus, because of the transcription, we have the RNA. Then RNA will move from nucleus to the cytoplasm. In the nucleus, proteins are going to be synthesized. And for example, if it is a muscle protein, these muscle proteins are all secreted out and then assembled, and then we form a muscle filament, in fact. I will not get too much details in this one. So now this is the whole process of finer structures of DNA, what is the RNA and what are the subunits. I will not get into that one. This must be very complicated genetic code. If anybody wants, probably I can explain it later, but right now we will just leave it at that particular point. Okay, so now okay. So we, since we do have RBCs, for example, And there is, for example, when you discuss about the sickle cell anemia, the difference we call this as a single nucleotide polymorphism. I think I will just explain that in this example. Now, this is an US athlete, fatalities due to mutation. So this was happened in 2008 in University of Central Florida. This has been cited in most of the textbook. Eric Plancher went into cardiac arrest after an off-season workout supervised by a coach, George O'Leary. He died a few hours later. Probably you also have read a lot more information that many athletes, while performing, especially enduring cross-country skiing, that is one of the endurance, and people collapse during the event, in fact. So now the question is, everybody will wonder, because before such event, everybody will go through a thorough medical checkup, and the medical checkup doctor says he is fit to carry out this event, but still he died. The question is that why he died, or are we missing something, is very critical, in fact. So the cause was understood, but everyone was very curious. Why did this healthy man suddenly die with no long-term symptoms or medical history? When we did the genetic analysis, this person is having sickle cell anemia. The sickle cell anemia trait can be identified only through genetic analysis, not by any other means, in fact. So later when they identified, so I will just give an example, what exactly is going to be happen. Okay, so now we have a normal, and then we have a mutated one, in fact. So usually what happens, like I will just go to that. Now we have a genetic code. 3 of the nucleotides are responsible for 1 amino acid. So I will just explain that one here. So now this is 1 codon, this is the 2nd codon, this is the 3rd codon. So usually what happens, so now that gives an amino acid, and these are all normal amino acids, results in the synthesis of a normal hemoglobin. So in case of this guy, that is Eric Planter, what happens is that, see, you see CCT, everything is same. Here GAG, here it is GTG. What happens is that this one nucleotide A is mutated to T. So now what happens? GAG will give glutamic acid, it's an amino acid, whereas GTG, because of 1, instead of a glutamic acid, it gives a totally a different amino acid value. But all of them are same. Rest of the protein in a hemoglobin, they are all same. But just because of 1 change, so now this will become a sickle-like structure. So the movement of these RBCs in the capillaries is hindered. So it is going to be blocked. So the binding capacity of oxygen is completely lost. So as a result, these people will get exhausted immediately. So such things will be noticed only at the higher stage. Probably you must be knowing that many of the, for example, if you want to measure the endurance, so we are going to have the treadmill test, we are going to have the cycling, we also have the rowing. These are all the equipments we are going to use. But the best one to understand the cardiac potency is going to be the treadmill test rather than bicycling or the rowing experiment, in fact. So because of the single nucleotide polymorphism, so this is the healthy one, this is sickle cell anemia. You can really see, I mean, how difficult it is to pass through such sickle cell anemia in the smaller capillaries. There will be a blockage that results in heart attack. So now this single nucleotide polymorphism is observed in many athletes. For example, now this is a muscle gene, ACTN3. So now sometimes what happens, because of the mutation, the muscle is going to be much stronger. So when you do see such mutation, you can always say he's going to be muscularly strong, so we can train him accordingly. So now this is the, so we do have a huge table. I can keep on telling that, I mean, how like a sickle cell anemia, we can discuss what are the genes and how these genes are going to be modified for the beneficial purposes, in fact. This is a gene in which I do a research in my laboratories that is called as an ACE gene. ACE gene is related to the hypertension, increased blood pressure. Okay, so now suppose if somebody is having a high blood pressure, so now if this gene is going to be blocked using a drug, so usually we have a lot more antihypertensive drugs. All these drugs will inhibit this ACE gene. Now, for example, by genetic, because if ACE gene is very active, then you will have the hypertension. What happens if the ACE gene itself is not active in a person? Then hypertension is going to be very rare. So with such person, we can train them for an endurance test. So this is the whole idea. So now some genes are very disease oriented. Suppose naturally these genes are all down regulated, they are really good one for our identification, so that we can train, because these anybody with a non-functional ACE gene can be a very good person for enduring sport, in fact. So I just give you a few examples like this. So now strength, endurance, speed, coordination, flexibilities. So you have a lot more events. For each one, I think there are set of genes are going to be associated with that. And a modification of something in those regions is going to be treated that, okay, this can be trained, because this itself is not enough. After that, this is only 66% what we are talking about. The rest of them is going to be the psychologically should be oriented, training should be done, proper guidance has to be done. So then those things will really help. So I told you that we have a whole lot of genetic markers. ACE1, this is for the endurance. We have many of the muscle genes. For endurance, I think we have identified a lot of genes, and many of those things are going to be useful in analyzing the whole process. So why detection? A basic genetic testing has been done for Eric Plancher. We could have trained him accordingly. We won't have put him into rigorous training. We could have informed that, okay, the child has got these particular complications. He can only do an exercise to a certain level. Beyond that, it's going to be very difficult. So if we had that genetic information, probably we could have saved the person. And like sickle cell anemia, we have the cardiac hypertrophy. So now we also given what is the prevalence, 3 in 100,000. So all these statistics are all easily available. So usually how the genetic test is done, we don't need blood. A cheek swab or a saliva sample is going to be done. Now in Delhi, I think there are some laboratories are already been conducting a sports gene test for 2,000 rupees, and more than 3,000 children across the people have gone and got this test done. They can claim that no, I have a sport gene, I can be a sport person, but whether it is really valid or not, I think you people have to tell us. So where exactly all this kind of studies has been established? It's been established in countries like US, China, Japan, most of the countries in Europe, UK, Poland, France, Spain. This also been observed in countries like Turkey, Israel, Uzbekistan, Lithuania, Brazil. Most of these studies are to refine the elite sport person. But India should establish, or we should think, at least in this line, I think basic groundwork has to be done, because the technology is very simple. If we can do per day lakhs together RT-PCR, so with the sports person, I think we can do that. I know that I think you people have access to a lot of national teams. So from those national teams, we can try to isolate the saliva sample, see all these genes. We can see that we can find out a correlation. Then we have to do that with the small children. If they do have this particular gene, then we can extrapolate the whole process, in fact. Is it feasible in India? But in India has become close to such methods for testing of doping and genetic variation still is not there. Athlete biological passport, which contains the athlete's genetic code, that's very important. Eligibility of the athlete in doping and gender by genetic based methods is all these things. I think we are still yet to get into the system, in fact. So methodology is going to be very simple: swab, then we isolate the DNA, sequencing, then with a computer we are going to find out whether it is good or bad. So much needed research we need, because already there is a template is been done in other countries. So we need to do that, but we need to have a sample study in Indian scenario. So now the genome-wide association studies, because again, I think I would like to stress that it's the single nucleated polymorphism is the one which we have to do that. So we need to have a case control studies and cross-sectional association studies. I think you have done a lot more research in this particular line. I think you have to design, and we have to form all those things. I think it's going to be the same thing again, saliva samples, and we are going to use the how we are going to do all those things. In case of a postgraduate degrees in sports medicine and sports sciences, I think we need to include this particular chapter. And we started to understand what is this genomic analysis. Positive aspects of this: it is less expensive, non-invasive. There won't be parents will not have any complaints. It definitely prevents death in case of there is any congenital anomalies. It builds an extensive ground for research and statistical analysis. And the ethics: with every great ideas comes its own share of alerts. A set of guidelines should always be followed for this testing. Seema Patel, I think, published an ethical stating that this method should not be misused to favor some children over the others. And no form of employment or sports qualifying test should be genetic based, because it is based on the performance impact, because this is an ethical issue which is very critical. It should be known that genetic basis of sports ability, though it is the ultimate core information of the person, it is not an absolute. The environmental factors, the psychology factors, the training, what you people give, they are also very important. 66% is not an absolute requirement. That is only a trend. Okay, you can be a person, but the remaining 33% will make the winning and losing of a person. This method should only be used to improve the training requirement, but this then cannot be used as an yardstick to judge in any other format. This is a very strong ethical issue. I think you all agree with this one. Scope: characterizing Indian indigenous alleles and trying to genetically determine potential athletes at a young age can be done, and appropriate training can be given at an early age. It is very crucial. App training regimes could be designed. Genetic screening may help determine which type of sports an athlete can be most suited. It has got wider scope for research, and this also covers both of the health and emotional status in this system, in fact. So in the conclusion, the entire concept, methodology, and idea is a form of optimization of sports ability screening. This helps to maintain the sports person's health, sports ability, and fitness to suit his or body requirement. This may nullify damage, helps in increasing the ability of the person, and can bring out the best in him or. The sportsperson of India are adored by the masses. They bring the nation honor and accolades and make their fellow citizens proud. To somehow perfect their training methods and make their health and fitness a priority by looking at the information they carry in their veins is one way giving back to them, in fact. I think I will conclude now. So physical activity and conjunctive decline, the role of the apolipoprotein E4 gene. So these are all the things, but we don't have a single gene, or we don't have a magic bullet to say that is good or is bad. So we need to analyze so many things in that regard. I think I thank Rushali Kamath, my MSc students who gathered almost information, and most of the things is in collaboration with Professor M. Chandrakumar. So if you do have any questions, I'm willing to help. I know that in some slides, it is very, very technical. If you have any questions in any of those slides, I'm more than happy to discuss. Thank you very much.
Thank you, sir.
Thank you, sir.
Thank you.
Thank you, sir.
All right,
Thank you.
Professor.
Any
Thank
questions?
you, sir. Thank you. Thank you, sir.
Sir, please share this PPT to all our email ID, sir.
Sure, because the whole thing is going to be recorded by the office. So now whatever the PPT is being done, it is in the laptop of the
MHRD.
staff college. Probably the organizers will definitely share all the slides. If you
Thank
want,
you so
I
much,
mean,
sir, for wonderful.
I will share this also with Madhyalagan. Through Madhyalagan, probably you should be able to get all those facts. No issues with that.
Okay.
sir.
Good.
Okay, sir.
Sir, just a minute.
Yeah.
Hello.
Go ahead,
Just a
sir.
minute, sir.
Thank you for the wonderful session. Now I request Dr. Radhakrishna, one who working as a physical education director at Government Degree College, Vamanapada. He is one of great athletic coach. He produces national and international athletes.
Good.
So I request Radhakrishna sir to please present a vote of thanks.
Okay.
Thank you, Rohit sir. Actually, it is a very enlightful session regarding doping in sports and talent identification. It's a very nice information, sir. Even though this COVID-19, as you know, physical education directors are very difficult to sit in the chair. But suppose it is a practical means, it is very good. Even though special thanks goes to Dr. B.S. Vishwanath sir for given very good information on behalf of HRDC Mysore and Department of Physical Education, Mysore University coordinators Dr. Madhyalagan and Dr. Venkatesh, and on behalf of all my colleagues in the various districts of Karnataka and Kerala, Tamil Nadu, and Goa also. Thank you, sir.
Thank you, sir.
one and all.
Thank you very much. Indeed, it was a pleasure.
Thank you, Vishwanath sir. Thank you, sir.
Thank you, sir.
Thank you, Radhakrishna sir, for your wonderful vote of thanks. Thank you.
Thank you, sir.
Thank you. Thank you. Lois.
Thank you very much.
Thank you. See you after.